can summarize this first part of the story as a focus on the development of electrical excitability, starting with calcium-dependent action potentials, moving on to understanding the underlying voltage-clamped currents, putting these into computer models of action potential development, identifying the role of potassium channel genes, and then finally imaging spontaneous activity, both in culture and in vivo. Well, of course, then one wants to know, what about that spontaneous activity? And so it was important to do both loss of function and gain of function experiments to try to find out what they might be doing. Here, I'll just summarize it for you. The take-home message was that the neurons in the embryonic neural tube during a 10-hour period generate this spontaneous calcium spike activity. This is what the embryo looks like at the start point, and this is what it looks like 10 hours later, generating these calcium spikes during that time. And the real interest was that suppressing activity led to a change in the numbers of neurons expressing excitatory and inhibitory neurotransmitters. So when activity is suppressed, the number of neurons expressing glutamate and acetylcholine goes up. And the number of neurons expressing inhibitory neurotransmitter, GABA and glycine, which are in fact inhibitory at this stage in this embryo, goes down. In contrast, when activity is enhanced, just the opposite occurs. Now we have more neurons making the excitatory transmitter, glutamate and acetylcholine, and fewer neurons making the inhibitory neurotransmitters, GABA and glycine. And you reflect on that for a moment and you think, goodness, that looks almost as though it's homeostatic. Because when activity is increased, the nervous system, when activity is suppressed, the nervous system responds by trying to boost up the activity that it generates. And when activity is enhanced, now the nervous system tries to damp down the activity that it's generating. So it looks as though it might be homeostatic. Well, this was intriguing for us, but right away it raises a question. During this time, neurons are making synapses with one another, and one has to wonder what's happening postsynaptically. If we change the identity of a neurotransmitter, and the neurons now formed a synapse with a postsynaptic partner or partners, if the receptor population doesn't change, we're out of business. The synapse won't be functional. So this was very interesting, and this developmental work then led us to a series of conclusions, which I want to just go through very briefly to wind up this part of the lecture. We think we've established 5 different features of transmitter switching during development in this model system. The first is that it is manifest in several different forms. It seems to involve, in some cases, the addition of a transmitter, as we just saw, dopamine to the neurons that already express neuropeptide Y, loss, as we'll see later on in the lecture, and switching of transmitters, in some cases, actually replacing one by another. So this is very intriguing. It seems to be homeostatic in the sense that it compensates for the change that's been imposed on the activity of the animal. We've been able to drive this by ion channel misexpression, but much more interesting and relevant, of course, is that it can also be driven by sensory stimulation. I'm not going to touch on it today, but we've had a close look at some of the molecular regulation here. This will come as no surprise to all of you that it involves regulation of the activity and expression of transcription factors. These are the DNA binding proteins, as we remember, that actually regulate gene expression. Importantly, the changes in the transmitter are coupled to corresponding changes in the postsynaptic receptor identity. This is obviously key. Without that, the synapse with the new transmitter won't work. And fundamentally, the most interesting thing probably is that this switch leads to changes in behavior. Well, I'll just summarize then the anatomical and molecular results of this part of the story, focusing on the adult nervous system. What we think we know now is that the suprachiasmatic nucleus that provides input to the paraventricular interneurons can drive their activity, we think, and as a function of the different photoperiods, can actually change the identity of the transmitter expressed in the cell body. We've looked also at the postsynaptic receptors, obviously, as you know now, a very important part of the equation. And interestingly, there is no change that we can detect in the red somatostatin receptor population on the target cells, these cells that release corticotrophin releasing factor into the cerebrospinal fluid in the third ventricle. However, there is, as diagrammed here, a very pronounced change in the expression of the D2 dopamine receptors. When the neurons are largely dopaminergic, these D2 dopamine receptors are there as presumably needed by the cells. And when dopamine is largely absent, these dopamine D2 receptors are very difficult now to detect. So at this point, one wonders, what about behavior? What's going on with the behavior here? So we interpret this to mean that there's actually a behavioral consequence of the new induction of these dopaminergic neurons. Well, this was fascinating to us, and we've begun to wonder now whether this could be related to a neurological disorder with which some of you will be familiar. This is not a disorder that we encounter very much here in San Diego, seasonal affective disorder. As you know, this is a consequence of exposure to very short, typically winter days, long nights, and people become very seriously depressed. And the intriguing finding reported here in 2006 was that in contrast to treatment pharmacologically with agents such as fluoxetine that were occasionally effective, and in fact often effective, but often had unwanted side effects and were ineffective in other cases, that phototherapy, 30 minutes of bright white light a day, was a very effective treatment for these patients. And so natural to wonder whether or not that could be involved in what's going on here. And this could, in principle, be very simply tested by PET scanning, looking for changes in D2 dopamine receptor expression. And this is on our dance card, something that we'd very much like to do. Well, try to put this all in perspective. I think we are all here very familiar with a variety of different forms of neuronal plasticity. I just draw quick attention to what I think of as classical plasticity, starting with changes in synaptic strength. This has been very well studied, indeed brilliantly studied by colleagues here on the campus. And for example, for long-term potentiation, Robert Malinow and his colleagues have, I think, really nailed this to the wall. I think it's very clear now that one starts with a synapse that expresses only one class of glutamatergic receptors, and then receptor trafficking introduces a second class, strengthens the synapse so that a single action potential, initially producing just a single action potential on the output side, now produces a series of action potentials, and the synapse has been strengthened. Changes in synapse number are also familiar to us. Changes in excitability through insertion of voltage-gated channels on the postsynaptic side, also very interesting. What we think we have here now with transmitter switching is what we're pleased to call, Davide and I are pleased to call reserve pool plasticity. We think that many neurons, and we don't yet know how many, I hope to find out, many neurons already express their transmitter, just as Darwin said. And they have a day job. They're wired up, they're doing stuff, and it's very important for us, and if we lose those neurons in a stroke, we're in big trouble. So there's no ambiguity about that. But we think that many of these neurons are also, in a sense, reserve pool neurons, in that they can acquire or lose a transmitter, they can switch a transmitter, they can change from a day job to a night job, they can change what they do. And actually, the first example of this that was suggested was in the mid-1970s, a switch of neurons in the superior cervical ganglion of neonatal rat pups. In culture, it was shown by a group at Harvard that you could switch from acetylcholine to norepinephrine and back again. Very interesting, but the tools were really not there at the time to run with this and go further with it and identify its role in behavior and its role in the adult nervous system. And what we've seen here today is that we can have switch between somatostatin and dopamine in this neurotransmitter switching process. So finally, in work that's ongoing now, just the last couple of slides here, we've begun to look at this plasticity as a function of age by simply taking rats beyond the 3-month period that I've been talking about so far this afternoon, 3 months of age. This is the data that we've been examining together, and look at animals at 12 months of age and then out at 18 months of age. And what we've done is to normalize the number of tyrosine immunoreactive neurons on a long day and on the short day to as a percent of those on the balanced day, 12-12 light dark cycle. And you've seen the data over here at the left before, very prominent change in the numbers of these neurons as a function of photoperiod at 3 months of age. 3 months of age, of course, would be a normal adult rat. But by 12 months of age, the ability to respond to the short day photoperiod with an increase seems to have been largely lost. And by 18 months of age, both directions of plasticity seem to have changed. So this is work ongoing. We haven't got this all figured out yet, but it seems as though there's an age dependence to this particular kind of neuronal plasticity, at least in the adult rat. In other work, Wei-Chan Lee in the lab has been studying the effect of exercise. And as you know, I'm sure mice love to run. We put them on a running wheel here, and they just run all night long. It's quite amazing. And what Wei-Chan has been doing is to look in the midbrain. We're very interested to know about other regions of the brain where transmitter switching might occur, and focusing on the pedunculopontine nucleus, the PPN, the pedunculopontine nucleus. What she finds is that this activity leads to an increase here with exercise of expression of C-Phos. What's C-Phos? C-Phos is an immediate early gene that's activity dependent. When your nervous system is active, more cells express C-Phos. So this is telling us that indeed some cells in this region are active. And in parallel, she finds a decrease in the numbers of neurons expressing choline acetyltransferase. So here with exercise, the number is down, and we look at the quantitative data here. We can see, yes, it's quite significant. And in contrast, the lateral dorsal tegmental nucleus, another cholinergic nucleus right next door, no significant change. So she's very interested in looking at the reversibility of this process, looking for a switch partner, and of course, most importantly, trying to identify what the effect of a decrease in cholinergic neurons here is on the motor behavior of the animal. In other work, Matt Eckler in the lab is looking at obesity, fundamentally, feeding mice on a high-fat diet and looking at changes in POMC expression in the hypothalamus. Now, POMC, as many of you know, is a large peptide. It's processed to give rise to a series of smaller peptides. Most interesting for the regulation of appetite is alpha MSH, alpha melanocyte stimulating hormone, alpha MSH. And what Matt finds, he puts the animals on a high-fat diet indicated in red and compares that to the food intake by mice put on the standard mouse chow. After about a week, we can see that the animals on the high-fat diet reduce their food intake, apparently balancing their energy efficiency, the amount of kilocalories that they take in. And then out at about 3 weeks, there's the weight gain begins. So we're very intrigued by what might be going on here at this very early point. And nicely, what Matt finds is when he looks at the numbers of POMC immunoreactive neurons in the arcuate nucleus down here at the base of the brain and the hypothalamus. Here's what we see under the control condition, but the high-fat diet leads to a very abrupt increase here, 1 week on the high-fat, and then this starts to decline again by 2 weeks and by 4 weeks, and the data indicate that this increase and then subsequent decrease are quite significant. So we're very interested in the same kinds of questions, the reversibility, the switch partners, the behavior. We think this is going to be a very interesting road to go down. So finally, where does this bring us? This shoots us up to the top right here, and looking at transmitter switching in the adult brain. Our objectives going forward into the future are to focus on both normal physiology, things like exercise, which we all indulge in, and also in neurological disorders, perhaps even psychiatric disorders. So that's the story. I think it's fair to say that the brain is ever changing. It's ever changing in the adult, not just during development. And I'm sure it's changing in ways that we have really not yet tumbled to. I think the transmitter switching process is fascinating. I think it has interesting implications for our interpretation of the connectome. Many of you know, many of us have colleagues who are embarked on the venture of mapping the connections within different nervous systems. Of course, most interestingly, perhaps our own nervous systems. This is clearly going to be very important for understanding how the brain functions. But it's going to be challenging to interpret the wiring if the wiring can stay in place, the axons and the dendrites and the synapses can remain in place, but the presynaptic transmitter can change and the postsynaptic receptor population can change as well to change the function of the cells. But there's a lot to do here, and I'm looking forward to doing it, and thanks very much for your attention.